Hello everybody, and welcome to today's presentation towards commercial manufacture of pluripotent stem cell derived therapies, presented by Rhys Macown, Senior Scientist at Cell and Gene Therapy Catapult. I'm Anne MacDonald of Thermo Fisher Scientific, and I'll be your moderator for today's presentation. Before we begin, I'd like to remind you that this event is interactive. We encourage you to participate by submitting any questions you may have at any time during this presentation. Just click on Ask a Question box located on the far left of your screen and type your question into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please use the Ask a Question box to let us know of any technical difficulties. So now please allow me to introduce Rhys Macown. Rhys Macown, Ph.D, is a senior scientist in the process industrialisation team at Cell and Gene Therapy Catapult, UK. Rhys earned a BE honours in chemical and process engineering at the University of Canterbury in New Zealand before starting his career with a Ph.D in biochemical engineering in Professor Nicolas Szita's group at University College London. There he designed and tested a microfluidic system for the 2D adherent culture of pluripotent stem cells with fine control over the microenvironment. Since joining Cell and Gene Therapy Catapult in 2014, Rhys has been continually active on fee-for-service, collaborative and core-funded projects developing pluripotent stem cell-based processes, and has played a leading role in exploring the 3D expansion of pluripotent stem cells as aggregates. So now I'll turn the presentation over to him.
Thank you, Anne, for the very kind introduction, and good morning, good afternoon, and good evening to everyone listening in around the world. As Anne said, I'll be presenting on our work towards the commercial manufacture of pluripotent stem cell derived therapies. But first, and briefly, I promise, there'll be a couple of slides on who the Cell and Gene Therapy Catapult are, particularly in relation to this talk. Then why we're interested in pluripotent stem cell derived therapies and major barriers to their commercial manufacture, before getting into what we've been doing to address these barriers and what our plans are going forward. So first, for anyone who's unfamiliar, who are the Cell and Gene Therapy Catapult? Cell and Gene Therapy Catapult is part of a world-leading network of technology and innovation centres. We were established in 2012 as a non-for-profit independent centre by the UK government through Innovate UK. We aim to accelerate commercialisation of research and growth of the cell and gene therapy industry through collaborative innovation, providing access to unique expertise and facilities, and working with policy and practice makers to create a fantastic UK environment in which the cell and gene therapy sector can flourish. Here in Guy's Hospital in London, we have established 1,200 square metres of state-of-the-art development laboratories. And we also have a dedicated viral vector industrialisation laboratory, which allows us to investigate the availability, cost and scale up of viral vectors. Slightly further north in Stevenage, we have built a large-scale GMP manufacturing centre, which allows companies to develop their large-scale GMP and commercial manufacturing processes in collaboration with Cell and Gene Therapy Catapult and benefiting from end-to-end expertise and practical support from our experts. And indeed, our most unique and important asset is the team of over 170 cell and gene therapy specialists based across those two locations, who between them have expertise in process industrialization, so that's my team, regulatory and clinical development, and various engagement activities. So one of our strategic priorities for the next 5 years, and indeed the most relevant to today's presentation, is intelligent manufacturing. Here we're working with therapeutic developers contract manufacturing and service organizations, and the supply chain to develop new technologies, intelligent systems and automation to increase productivity and reduce cost of goods, so as to confer both technological and cost advantages to industry and to reduce the reliance on labor-intensive processes. So this schematic shows one way of illustrating this approach by presenting 4 progressive phases of automation. We start on the left with manual processes, which are typically well established, but open, labor-intensive, operator-dependent, and generally higher risk. The first step towards automation involves using multiple pieces of automated equipment in sequence to deliver the various processing unit operations applicable to the process in question. And this should result in a closed process with increased reproducibility and robustness, and also provides the opportunity to bolt on process control systems with the same modular approach. The next step is to link pieces of equipment together into an integrated system, resulting in increased containment and efficiency, reduced labor, and allowing for a more integrated process control strategy. And finally, we see that there's an opportunity for a step change from that integrated approach of manufacturing a single batch to high throughput systems and predictive process control, and in doing that, further decreasing the footprint and reducing costs. So the key message and objective here is that as processes become more automated, the cost of goods are reduced and the potential of industrialization is increased. Okay, so that's where today's topic comes in. Why pluripotent stem cell derived therapies? Well, as we all know, all cell or gene-modified cell therapies can be split into one of 2 categories, autologous and allogeneic. Autologous therapies, you use your own cells, which are taken in a biopsy or a blood collection, manipulated through selection, expansion, differentiation, and/or gene modification, and then administered back to you. Each patient is their own cell source, and each therapy for each patient is produced in parallel. And because it's their own cells, the patient's immune system should accept the cells without any immune rejection. In contrast, in an allogeneic therapy, the cells come from somebody else. They're again manipulated through selection, expansion, differentiation, and/or gene modification, and then administered to multiple in patients. Because they're from someone else, you can use the same source for multiple patients. You can make product for all of them in the same batch. However, because they're someone else's cells, there is the increased possibility of the product cells and the patient cells having an immune response and attacking each other. So in contrast to autologous therapies, allogeneic therapies can be manufactured in advance instead of once the patient needs them. This means they can be made available off the shelf and administered as soon as the need arises, which is particularly important for trauma or acute conditions like acute myocardial infarction or stroke, for example. Advanced manufacturing also means that failure of a batch is less likely to directly impact patients relative to an autologous equivalent, where it may not be possible to complete a new batch. And furthermore, allogeneic therapies could also be less invasive to patients than autologous equivalents, as no collection of starting material from the patient is required. It could be important considering the patient is very unwell and may struggle to endure the collection process. And then, more from the process side, because the batches aren't patient-specific, larger batches should be possible. Introduces the potential to use established biopharma technologies such as stirred tank reactors, possibility to scale up rather than scale out processes, to introduce lower cost of goods through economies of scale. So to finish my case for allogeneic therapies, I'd like to consider a generic autologous therapy example. In our experience, autologous therapies take from less than 7 to over 60 days to produce, but let's assume this one is 10 days long. Let's assume that the process fits on a compact all-in-one system with a busy day at the start when you receive your patient material, a busy day at the end when you formulate and fill your product. Well, at 10 days processing time per batch and allowing some turnaround time between batches, you should be able to produce 18 batches per unit per year. Now, if we take a market size that was estimated for one of our projects, we would require more than 190 parallel units to produce this product to meet market demand. And if we assume that each suite in our new manufacturing center could support about 16 units in parallel, we would need one full manufacturing center to make this one product. So Google Maps was not updated when I downloaded this picture, but that blue box is the rough footprint of our manufacturing center located at Stevenage next to the Bioscience Catalyst and the DSK site. However, if we consider a larger indication, another project we worked on estimated a market size of approximately 35,000 patients a year. So now we're talking greater than 1,900 parallel systems and approximately 10 manufacturing centers to produce a single product. So we've become quite a large occupier of space at Stevenage. In contrast, our estimates for an optimized allogeneic process for that project predicted you would need approximately half of one of our manufacturing centers to manufacture for that market size. It just shows what sort of contrast you can get in the scale and the space required to deliver the same market. Okay? So within Allogeneic, why develop pluripotent stem cell derived therapies? Potential sources of cells for allogeneic therapy include somatic cells such as mesenchymal stem cells, cells like T cell modified ex vivo to reduce the risk of immune response, immortalized cells, and the pluripotent stem cells that we have chosen as the exemplar to begin our development. Pluripotent stem cells, by definition, can differentiate, produce almost any cell type of the adult body. They can also divide indefinitely to produce more and more pluripotent cells. And both 2D and 3D processes have been demonstrated for pluripotent stem cell expansion and differentiation. And it's this combination of properties that give pluripotent stem cells their immense potential as starting material for cell therapies, and which drove our choice as to where to start our development work. Now, having listened to my case for them, you may be asking, why aren't we all already using pluripotent stem cell derived therapies? Indeed, in our non-clinical and clinical trial reports for the UK, so non-clinical on the preclinical on the left and clinical trials on the right, allogeneic therapies make up approximately half of preclinical trials and just 1/3 of clinical trials. Why is that? Well, first of all, autologous therapies sometimes do make sense, particularly where the market size is small, shelf life is low, and the risk of immune rejection is high. But further to the advantages we've already discussed, allogeneic therapies also face some significant challenges. So first and foremost, safety. As mentioned earlier, there is potential for immune response. Different groups around the world are currently working to mitigate this through HLA matching, by donor selection, so universal donors, or cell line engineering to create immune-evasive cells. And depending on the therapy, the mechanism of action is not always well understood, which can present challenges setting up a target product profile. And there's also always the concern at the potential for tumorigenicity, particularly when starting with cells self-renew indefinitely and when expanding cells for extensive periods in vitro. But then looking at the manufacturing processes, current processes are often open, manual, involve animal products and serum, and have limited assays established for monitoring and control. These factors make processes inefficient and unreliable, which is to say expensive, challenging from a regulatory point of view, and impractical to scale up to meet market demand. Just to put things back in perspective, remember that when pluripotent stem cells have been grown historically, within dishes and flasks, on coatings made from animal products whose composition wasn't known and was variable, in culture media containing serum, who again the composition wasn't necessarily known and was variable, and when they ran out of space in their plate or in their flask, they were passaged into the next by manually scraping to generate clumps of just the right size. So not what you would call an industrializable process. And unfortunately, the scale required is rather large. But even assuming that the relevant differentiation process can achieve a yield of 100%, the manufacturing volume to expand these pluripotent stem cells is very large. So there is a clear need for robust, efficient, scalable processes to manufacture to meet market demand. So that's my little sales pitch. So what are we here at Cell and Gene Therapy Catapult doing about it? Well, this is the strategy we've taken in setting out to address them by developing those closed, automated, efficient, and scalable platform processes. Our strategy includes the generation of embryonic and induced pluripotent stem cell banks adapted to defined industry standard culture systems and characterized to industry standards. Exploring parameters to increase the efficiency of both 2D and 3D pluripotent stem cell culture. Scaling up both 2D and 3D PSC culture. Investigating systems to harvest, concentrate, and wash those pluripotent stem cells. Underpinning all of that, developing an analytical framework of relevant assays and measurements that can analyze and direct our process. The idea is all of this will result in platform processes, assays, and development approaches that can be leveraged for different pluripotent stem cell derived therapies. So we work with two exemplar sources of pluripotent stem cells, the Chef 6.1 line, a human embryonic stem cell line kindly donated by the UK Stem Cell Bank, and our RCIB 10 line, an IPS line that was the pre-seeded lot of which was derived under GMP conditions, but it is a research grade bank. And the research grade IPS bank has been distributed to external collaborators globally who have collectively demonstrated differentiation potential to cells of all three germ layers. In-house, we have tested more than 10 reagent systems for 2D PSC expansion. We have characterized cell banks adapted from either the human embryonic or the induced pluripotent starting material. And we've generated protocols for standard 2D culture and analytical assays for quality testing and characterization in terms of both safety and identity. So here you see examples across the top right of morphology and growth rate, in the middle row of phenotype by flow cytometry and immunostaining. Differentiation to the three germ layers is shown in the bottom left by immunostaining. And on the bottom row, we also have gene expression profiles and measures of karyoology. So this is the level of characterization we're applying to those banks that we'll generate. Then looking at 2D expansion, we have explored parameters for 2D culture ourselves and through a number of collaborations, investigating different culture media, passaging agents, and extracellular matrices. And when we do these investigations, again, we investigate growth rates and viability, morphology, metabolism, protein expression by flow, and gene expression by qPCR. So here in the top right, we have a figure comparing growth in 6 different media. And on the bottom row, we have a study of different combinations of media and 2D coating material, again showing growth rate, morphology, and expression of pluripotency markers by flow cytometry. We have also investigated different medium exchange regimens. So these two figures are from the same experiment. The bottom right shows the concentration of metabolic waste product at the end of each day of culture. And as you can see, there's relatively little difference between the different feeding regimens. However, when you look at the top plot, which shows viability and viable cell density, you can see that there's a very significant difference in the final viable cell density achieved with these different regimens. Then when it comes to 2D scale up, our approach has focused on a hollow fiber bioreactor, the Quantum from Terumo. In the hollow fiber system, the cells grow inside the wall of thousands of tiny fibers, and medium can be circulated both inside and outside, with small molecules able to diffuse across the wall of those fibers. Now this provides 2.1 square meters of culture surface in a system with a relatively small footprint. But I think more importantly, the hollow fiber system allows you to continuously replace medium and control gas exchange, resulting in a more controlled culture environment for your cells. A challenge with the hollow fiber system is achieving high cell densities and recovering the cells at those high cell densities. However, we've found that we were able to seed at lower densities than we typically use in flasks, and we're still able to achieve similar fold expansion in cell number, which we see here. So metabolic measurements have allowed us to predict the cell number relatively accurately in a system where it's not possible to image or sacrifice wells, for example. So this plot on the left, the hollow triangles are showing the predicted cell yield based on metabolic measurements, and then the solid triangle at the end was the actual harvest yield. We've developed a process shown here where population doublings, cell viability, and media consumption are reproducible and favorable in comparison with flask-based culture, as you see in the two top right plots. And our experiments so far indicate that using the hollow fiber relative to 2D plasticware can reduce cost of goods by 45%, reduce labor by 2/3, and increase facility throughput by 250%. You can see it's not just about closing and automating, but you do get significant returns in terms of savings and throughput. Then looking at the 3D side, our initial approach to 3D expansion has been to investigate aggregate-based expansion from single-cell seeding with periodic settling to allow for the medium exchange. So you see here across the bottom some examples of expanding pluripotent stem cell aggregates over the course of an experiment. The process parameters we initially explored were explored at 15 mil scale on the AMBER 15 system, pictured top right. So this has either 24 or 48 stir tank reactors operating in parallel. And at 150 mil scale in the DASBox system, pictured bottom right, which has 4 parallel STR units per controller system. And the parameters we've explored are impeller speeds, feeding regimen, and starting cell concentration, again while measuring viability and growth, aggregate size, metabolites, and expression of pluripotent markers. So in the top right on the slide, we have a figure showing viability and viable cell density with 3 different feeding regimens. And again, you can see the feeding regimen really has a significant impact on the final viable cell density achieved. And then in the bottom right, we have a contour plot of maximum population doublings achieved for different impeller speeds and seeding densities. Now that plots from a model fit to experiments conducted at different combinations of impeller speed and seeding density. So that's helping us narrow down the design space in which we want to work. And then here, a couple more figures. So the top right is the profiles of 2 different metabolites, again in the experiment investigating different medium exchange regimen. And in the bottom right, we have an example of image segmentation of a picture of our aggregates. So we regularly take samples and image the aggregates and then use image analysis to determine the aggregate mean size and distribution. So we have used the results of small-scale experiments and engineering parameters for different vessels to scale the expansion first to 150 mils and then up to 1.5 litre scale. And in doing so, we've demonstrated the scalability of an aggregate-based expansion process from 15 mils to 1.5 litres, as shown in the bottom left. And we've achieved over 3 billion cells at the 1.5 litre scale. The cells produce, maintain expression of pluripotency markers by flow cytometry and still demonstrate the potential to differentiate into the 3 germ layers. So that's shown here with staining for markers of each of the 3 germ layers. And again, our initial expansion process achieves similar population doublings to 2D flask culture with reduced median consumption per cell produced. And cost of goods modelling here indicates that this SDR process would again deliver significant reductions in labour and cost per billion cells, as well as a significant increase in your facility throughput. Oh, I had an animation there. However, we feel that there's the potential to further reduce those costs and increase those throughputs through intensification, as well as potential to close and automate the whole 3D expansion process. So we've begun investigating different cell retention systems, both to enable closed and automated cell passage and to facilitate medium exchange. So on the right are some images of a trial we did dissociating aggregates in the vessel at the end of an expansion, then replacing the dissociation agent with fresh media in a closed and automated way using a TFF approach. And we were then able to get yields after passage comparable to our reference material, our reference process of manual centrifugation. Okay. We have been testing other technologies for concentration and wash, whether it be to remove the passaging agent, as shown there, or changing from expansion to a differentiation medium, or before and after other processing steps, such as a selection process or transduction. I'm going to talk about the fluidized bed centrifugation technology, KSET, and LOVO, a spinning membrane filtration technology. But there are other technologies in our investigation pipeline, such as TFF and acoustic filtration, for example. So KSET is a fluidized bed centrifuge. Liquid is pumped towards the center of a spinning rotor, and cells are trapped in a fluidized bed where the opposing forces of the fluid flow and the centrifuge are balanced. So to improve recoveries and viabilities using this equipment, we have investigated the bed formation flow rate and bed formation time, shown in the top right, as well as the process flow rate, shown in the bottom right, and the wash flow rate. And so far with the KSET, we've achieved recoveries greater than 80% when concentrating and washing pluripotent stem cells. The LOVO, on the other hand, uses the spinning membrane technology. So it cycles cells past a rapidly spinning cylindrical membrane, and it takes advantage of the resulting hydrodynamics to help prevent filter clogging. And among the parameters we've been investigating here are the filter spin rate and the starting concentration of cells in the feed material. So we have, again, two heat maps shown on the right here, one for cell viability and one for cell recovery. And so far, again, with the LOVO, we've achieved recoveries greater than 80%. So what impact does this all have? Well, these two figures are from failure mode and effect analysis, an exercise in which a group of subject matter experts systematically identify and score risks based on severity of impact, likelihood of occurrence, and probability of detecting the problem. The plot on the left is for a completely manual process, so 2D culture in flasks, followed by 3D culture in shake flasks. The figure on the right is for an automated process utilizing a hollow fiber system, followed by a stir tank reactor with harvest and wash using a fluidized bed centrifuge. So you can see that while some risks remain, the number is greatly reduced, especially of the high to critical category. So these are things that are very likely to go wrong and very likely to have a severe impact on your process. And particularly down here, where we get into the passage at the end of the 2D culture, the 3D expansion, and the harvest, concentrate, and wash, which are the areas that are particularly addressed by the slides I've just been through. So this is just to emphasize what large impact these automating and closing activities can have on the risk profile of your process. Now, in addition to all the process development I've talked about, running in parallel, we've been developing a new analytical framework. And a key objective of this has been to reduce quality assessments based on operated judgment, because they're so hard to replicate and tech transfer, and it's so reliant on having and retaining key skilled staff members. So to that end, we've been implementing image analysis algorithms to extract confluence and texture from images of 2D cultures, which I'm sure most of the listeners are familiar to doing themselves, as well as to extract the aggregate size distribution from 3D cultures. And here on the right, some illustrations showing how an entropy approach makes calculation of confluence much easier than just doing a brightness threshold. And you can see at the bottom there how the algorithm has picked out the outline of our 3D cultures very nicely. And then the reproducible and quantifiable metrics, such as the outputs of these algorithms, can then be compared with other outputs, which is what the bottom right image is illustrating, such as flow cytometry, cell density, to identify target profiles for different processes, which then feeds into a monitoring and control strategy. Another piece of analytical development that we've been doing is developing a more cost-effective qPCR assay than the commercial kit we had been using. So our in-house assay uses slightly fewer genes and much smaller sample volumes than the commercial kit we had been using, but delivers similar gene expression profiles across all those genes where it overlaps. And then again, on the analytical side, we've been analyzing the concentration of different metabolites and medium components by a variety of techniques, which include LC-MS and Raman spectroscopy. So here on the top right, we have a figure showing the stability of a particular medium component at 3 different temperatures, which informs on how long you can have it sitting out on the bench, for example, before you add it to your system, and then how long it's going to be present in your culture. On the bottom right, we have the concentration of a molecule at the end of each day of culture across 3 different passages. And in the bottom left, we have the concentration of a molecule after 6 days of 4 different processes. So you've got APSC expansion, directed differentiation to ectoderm, to endoderm, and to mesoderm. correlation of different soluble compounds with process parameters and outcomes, we think really offers a great potential to identify novel, non-invasive methods of process control. So I'm going to move now from what we've been doing to what we want to be doing going forward. If I don't jump in the slides, there we go. So our goals in the next couple of years include further intensification of the 3D expansion process. That's something I touched upon. We want to keep increasing the yields we can achieve using 3D expansion processes. We want to demonstrate further scale up of both the current and intensified expansion processes. So we've made scaled up to 1.5 litres so far, but we would like to keep scaling further. We want to translate exemplar differentiation processes to the 2D and 3D closed and automated systems that we've discussed here through expansion. And then in terms of enabling real-time monitoring and adaptive control, we want to further characterize PSC expansion and differentiation processes. We want to identify and develop assays for surrogate quality markers. So that was what I was just referring to, for example, with the measures of metabolites. And we want to bring the focus onto that early germline differentiation, which will be the most universally translatable to any process that a collaborator might come to us with. Finally, we're assessing novel materials and technologies to increase productivity and reduce the cost of large-scale processing. An example here, different novel microcarrier technologies. And the end goal, again, just to reiterate, is processes, assays, and development approaches that can be leveraged for any PSC-derived products to deliver a controlled and reproducible, and automated and scalable process that can get people's products through to late-phase clinical trials and commercial manufacture. Okay, so we'll wrap up with thank yous. A big thank you to all of the team members who were involved in producing the work I've shown to you today, and to all of the different companies and institutions we've collaborated with over the past 5 years. A big thank you to Thermo Fisher for inviting me to talk to you all today, and finally thank you to everyone for listening in. That's me.
So thanks, Rhys, for that informative presentation. So we're now going to begin the live question and answer portion of the webinar. So if you've still got a question you'd like to ask, please do so now by clicking on the Ask a Question drop-down box located in the far left of your presentation window. Type your question in the box that appears on your screen and click the Send button. Now, we've already had a number of questions already. So our first question is, when will stem cell therapy be actually available as a treatment to patients?
So, I mean, there are already cell therapies on the market. There are stem cell therapies in clinical trials, famously for retinal disease. So, I mean, my impression is that we're not far away from seeing them start to roll out into market. I think the first players will be for smaller indications, and the eye is a particularly attractive target because it's immune privileged. I think for larger scale, larger markets and less immune privileged indications, probably looking at 2 to 3 years from my perspective before we start seeing them really grow in the market. But they are there in clinical trials now, and it's just a question of how quickly those clinical trials can find patients, and how quickly we can address these processing issues that I've discussed today. And what we're trying to do from the process industrialization point of view is make sure that it's not the processing that's the limiting factor to getting them to market.
Thanks. So we have a question which I think relates to early on in your presentation, where you were discussing towards intelligent manufacturing, where you had mentioned that for manual processing was open but also high risk. And so we've got a question which says, what do you mean by high risk exactly? Can you please elaborate a little bit more?
Yeah, okay. So obviously, if we have an open process, it is a higher, it's not guaranteed, but it is a higher risk of contamination, which is something we all want to avoid. But also in terms of manual processing, because it's a person doing it, you're always running the risk of variation, both operator to operator, and even variation in that operator's performance day to day and minute to minute. And we still see processes that have scraping or pipetting at just the right speed and just the right number of times. Processes where you have to achieve a uniform cell suspension by manually mixing. And it's very, very easy to introduce a lot of variability there, which then, depending on where it is in the process, translates to product variability, and whether or not that variability is acceptable to pass on to the patient is going to be specific to the process step in therapy. But just getting away from operator judgment and operator dependent steps will really reduce the risk of your process being unsafe or ineffective.
So lots of more questions coming in here. So let me just go back and so someone's also asked about you mentioned allogeneic and they were looking for a little bit more understanding of what does that mean. Does allogeneic refer to the stem cell or any cell that will be later used to modify into the pluripotent stem cell? So I guess they mean the donor cell that is reprogrammed. So perhaps you could just do a little definition of allo versus auto.
Yeah, so autologous is where the starting cell material comes from the eventual patient. So the cells might just be purified or might be expanded. They might have gene modification, so they present a particular surface protein. But the key thing is that the starting cell has come from the patient, and therefore the HLA type and all of those proteins involved in potential immune response should match the recipient and shouldn't prompt a immune response. Whereas, as I said, allogeneic, you have a donor material, and typically it's a cell bank. Theoretically, it could be primary cells from someone else. But the cells have come from a different person to the patient. I hope that makes it a bit clearer.
Absolutely. And then related to that, we have a question around in allogeneic iPSC therapies, would there be more chances of immunologic responses? And so how can we overcome that? Again, you touched a little bit on that in your presentation, but if you could expand a little. Yeah,
I'm not sure if the the question meant specifically IPS or pluripotent in general. Obviously, because the pluripotent stem cells aren't from the patient, there is an increased risk of immune response. It's not something that we're heavily involved in addressing, but we are aware that a lot of groups are trying to address this around the world. The two main approaches that I'm aware of are selecting donors with an HLA type that will be accepted by a large portion of the population. So this is similar to being O negative in blood donations. You know, anyone can receive O negative blood. If we can find donors who have an HLA type that would be accepted by a large percentage of the population, then we could make therapies from a small subset of donor materials and be in a position to treat the majority of patients. The other approach that I'm aware of is genetically modifying cells so that we either achieve that HLA type, which will be highly accepted, or remove the surface proteins that are most likely to cause problems altogether, again creating a cell material that should be accepted by the majority of patients. And I know different groups are pursuing either or both of those two avenues. I think both have strong potential. I wouldn't like to put all my eggs in either basket at this point.
So a couple of technical questions now. For the immunohistochemistry, can you let us know what markers you're using to test for endoderm?
So I'm not a member of the analytical team. I don't know the full panel. So this slide here was just showing a single marker, and I think the earlier one was as well. So in this case, for endoderm, we were looking at SOX17, I believe it was.
And then a similar question was what cell lines should I choose for checking neural genes expression?
Sorry, could you repeat that one?
What cell lines should I use for checking neural genes expression? I wonder whether that's also what markers should you be looking for neural gene expression.
Yeah, I wouldn't like to answer off the top of my head.
Okay. Coming
from an engineering background, that's something I would always go and research myself.
Okay, so let me bounce back to an engineering question. We have a comment asking about is there any way to look at online monitoring of things like cell numbers, or do you look at other types of online monitoring as these cells are being expanded?
Yeah. So depending on the system they're in, different things are available to us. I showed in the case of the hollow fiber expansion that we were monitoring metabolites that's currently at line rather than online. In terms of the 3D systems, obviously STRs widely used and are designed for multiple probes. So we have your standard pH, DO, and temperature measurements, but we've also had Raman probes in there, which is a nice online system. And then there are different automated sampling systems which can get you close to online depending on your assay in question. So obviously a viable cell count of single cells is something quite quick. So an automated sampling system can withdraw a sample and count single cells. If it requires a dissociation step in between, that will make it more complex. but certainly we want to include online monitoring, and through the current at-line and offline monitoring, we're trying to prioritize what sort of probes we want to put in there. For example, there'd be no point looking at a metabolite that doesn't help inform the process. But if there's something that can tell us that, oh, you need to ramp up your medium addition, or you need to add more of this particular growth factor sooner than you would normally, purely by reading a metabolite or a cytokine, then that would be ideal.
Oh, thank you. Another question actually was, what type of cells are you using? But I think we answered that in the presentation. You have your 2 test cell lines, one, the ES line from Sheffield, the chef line, and the
other one
then
was
you
the
also had an IPS line that you made in-house.
Yes.
Okay. Someone's asking about, what kind of tests do you have to do to ensure clinical trial safety? Is that something that you're involved in or aware of?
So we're not, that I can think of, currently making any PSC-derived stuff ourselves for clinical trials, but we're involved in collaborators who are in clinical trials. It's always, as with all these things, it's always a conversation with the regulator, and I was careful earlier in the presentation to say that a lot of these less desirable attributes of processes make things regulatorily challenging, because if you can justify the need and demonstrate appropriate controls and safety studies, then you can always make a case to the regulator. I believe there will always be a requirement to show you haven't got detectable levels of pluripotent cells remaining in your differentiated products, and the regulators have made suggestions on what markers they're looking at for there. For example, the OP34, they want to show that you haven't got cells still expressing OP4 in your final product. Depending on whether your cells have been inactivated or not, there'll be higher or lower requirements there. Certainly for active cells, the karyology is going to be pretty critical. But as I say, it's very hard to draw up a list for therapy without knowing what it is and what process it's been through. We always would encourage people to get regulatory advice throughout their development. And
so have a conversation with the regulator as early as possible.
Definitely, and really try and start thinking about what assays you'll need for your commercial process before you need them, because the requirements will ramp up as you progress through clinical trials. And you don't want to get to the end of phase 3 and suddenly need to develop some assays and have to do more and more runs to do your assay development. You want to be developing that in parallel with your clinical trials so you can use that material for your analytical development.
Okay, we have a question a little bit sort of future focused of, can you see a future where an active organ might be completely made by stem cell technology?
I would love to see that future. I think that would be an incredible achievement on behalf of the whole industry, and then that's sort of a moon landing moment if we're recreating whole complex organs. I know there are exciting innovations in the field, and it's often often not mentioned because it's seen as further from the market, and it is further from the market because it's a more complex endeavor to both produce the multiple cell types present in an organ and then the structure around it. It's gonna take us longer to get there. I hope we can. Obviously, shortage of donor organs is a major issue, and I would love to be able to see us get to that point, and even to be involved in it. But it's further away than therapies based on a single less structured cell type.
So you mentioned the need to remove pluripotent stem cell from the final product, and we have a question here about, is there any risk of oncogenesis associated with PSC-derived allogeneic therapies? And I guess the answer is yes, which is why the regulators want to make sure that you remove all of the PSC cells from your final product.
Yeah, there is, and I raised it as one of the barriers. It's not something that's entirely processing technology related. It's also the process you're going through. Obviously, if you're starting with a cell line that can self-renew indefinitely and produce almost any cell type, then if you put those cells into a person, then there's a chance they can do just that, which we don't want. And similarly, depending on how much in vitro culture is involved in your process, so the extent of expansion and differentiation, and also how different the conditions for that are from what would occur in utero, these things can increase the likelihood of changes and mutations that would result in a malignancy. So depending on again whether your product has been inactivated or not, there will be differing requirements to screen your end product against the risk of tumorigenicity.
Okay, yeah. Thank you. Unfortunately, we're not going to have time to cover all the questions that have been sent, but just one more. Given all of the work that you've been doing with all these different processes, do you publish those protocols either through peer-reviewed journals or on the Catapult website so that people can see the types of things that you've been developing?
I don't think we have published many if any of them. We're working on publishing in journals, which would then obviously have methodology sections in it. And when people approach us to collaborate, be it on a grant or through a fee-for-service arrangement or whatever arrangement is worked out with our business development team, that can include access to the protocols we've developed. Obviously, we like to keep hold of some know-how, and I will apologise because I've been the frustrated person on the other side of the presentation. I do apologise that I anonymised a lot of the figures presented and removed numbers and cell lines and conditions, but obviously we want to hang on to some know-how that we can then share it with collaborators. So we're not trying to make it impossible for anyone to use it. We just would like to do so through some form of collaboration. But yeah, as I said, we are working to publish in journals, and when we do that, the methods involved in what we've been doing will be part of those publications.
Thank you. So once again, I'd really like to thank Rhys for his presentation. Before we go, please note that today's webinar will be available for on-demand viewing through to February 2019. So we can always go back and review the talk. So thank you again, everyone, for joining us. Thank you, Rhys. And thank you to all the participants who asked questions. Sorry we couldn't get through everything, but thank you very much. We hope you enjoy the rest of today's event.
Thank you, everyone, and have a great day, evening, night, wherever you are.